Okay, so let's get started. So, hi everyone, and thank you very much for joining us this evening for what promises to be a fantastic discussion. My name is Dr. Abbas Tejani. I'm a GP partner and occupational physician based in Leicester. I wear a number of educational hats, including being chair of the City Academy at the Leicester Medical School, as well as being the first five chair for the Royal College of GPs in Leicestershire. As some of you may know, I've been at the forefront of postgraduate medical education in Leicestershire for the last 5 years or so, and I've recently been appointed as an honorary fellow at the Leicester Medical School. I'm co-founder and director of Cornerstone Medical Education, along with my wife, Dr. Israel Tai, and we host a vast number of courses up and down the country, from subject masterclasses, COVID and the vaccine being one good example, to practical courses such as minor surgery, pessary insertion workshops, proctoscopy, you name it, we provide it. Our next two-day minor surgery course in Cardiff in December is now fully booked, but we have one running in Manchester on the 13th and 14th of March, as well as another one running on the 2nd and 3rd of July in Edinburgh. We also have a one-day minor surgery update course running in Leicester on Friday, the 26th of February, because as you know, to continue practicing minor surgery, you need to attend a minor surgery update course every 3 years. So if any of these courses are of interest to you, please visit our website, which you will have done to sign up to this particular webinar, or drop me a line and I can share more information with you. Now, one of the other things we're really passionate about is retention and recruitment in general practice. We've experienced firsthand how difficult it is to recruit in primary care. And at the same time, we had lots and lots of friends approaching us looking for jobs in primary care. So we decided to create a platform called GP Jobs, bringing the two needs together. So the website for this is gp-jobs.co.uk, and I'll put that again in the chat in a few seconds. So we're not an agency. There are no middlemen. In fact, no agencies are even allowed to sign up to our platform. Only NHS registered practices for people looking for jobs in primary care, from doctor to nurse to allied health professional, administrative staff, reception staff. It's free to sign up and look at what sort of jobs are available. And once you set up your profile, you'll get an alert each time a job is posted that matches your profile criteria. And each practice that signs up gets a free job advert to use, and thereafter it's a very small fee which you pay per advert. There are no hidden fees, no ulterior motives. We're just two GPs interested in retention and recruitment and want to help our specialty continue to thrive. And so because of that, we're one of the fastest growing and most innovative recruitment platforms. So come and sign up. You've got nothing to lose, and I'll be dropping the link for GP Jobs in the Cornerstone Medical in the chat function shortly. So I am absolutely thrilled and delighted to be joined once again by Dr. Julian Tang, who's a consultant virologist at the University Hospitals of Leicester and an honorary associate professor in clinical microbiology at the University of Leicester. Now, something that I didn't know, but that I've come to know over the last few months, is actually Dr. Tang trained in medicine and zoology at Cambridge before completing a zoology PhD in biological fluid dynamics up in Aberdeen. He then finished his medical training in Sheffield, and after general medical training, he completed his specialist clinical virology training at the University College London in 2005. Later the same year, he moved to Hong Kong after the SARS 2003 outbreaks as an assistant professor, developing a clinical and research interest in respiratory viruses, particularly on influenza and its transmission. He then moved to Singapore in 2008 as a consultant virologist, arriving there in time for the 2009 H1N1 influenza pandemic. And there he built a 1M diameter Schlieren imaging system to visualize human exhaled airflow, such as breathing, talking, coughing, sneezing, singing, to help aid aerosol infection control guidance. And after several years in Singapore, he then spent a couple of years working up in Edmonton, in Alberta, in Canada, where he described the first imported fatal human case of H5N1 avian or bird flu into North America in 2013. And then he returned to the UK in 2014, settling in, of all places in the UK, in Leicester, where he has been running the diagnostic virology laboratory and advising on the clinical management and infection control of viral infections, particularly respiratory viruses like the influenza and SARS-CoV-2, which is currently causing the COVID-19 pandemic. So someone who's really been at the forefront in tackling all the major pandemics in our lifetime. So we're absolutely delighted that Dr. Tang is with us today. And someone that I was speaking to actually advised me that Dr. Tang is probably one of only 6 leading virologists in the entire world at the forefront when it comes to COVID-19. So we really are very delighted that he's with us, and he's actually one of my patients as well. So before we kick off, just a bit of housekeeping. We're expecting up to 300 people that have signed up. So the plan is for Dr. Tang to go through his slides from start to finish. But if you have any questions along the way, please ask them in the chat section of your screens, and I'll pick them up and pose them to him at the end of his slide set. And after the Q&A with Dr. Tang, we'll have representatives from the Leicester City and West Leicestershire CCGs. We'll be talking through the practical implications for GPs in Leicestershire and Rutland. Throughout the duration of the talks, I'd be really grateful if you'd kindly keep your microphones on mute to prevent any disturbance as we go along. So there's lots to get through, and without further ado, I'm going to hand over to Dr. Julian Tang. I think you're on mute, Julian. There we go.
Okay, I've got a few problems with my internet connection tonight. I'm not quite sure why, but let's just see how it. So anyway, thanks to Abbas for inviting me again to give you this update. I've given a few of them over the past few months, and tonight we're going to focus on the vaccine. Now, it's been a very exciting couple of weeks with all these vaccines coming out and lots of data coming at you from all sides. And what I'm going to try and do is explain what the data means and how they get to certain conclusions about efficacy, so that you can follow these figures coming out in the next few months, because there's several other vaccines coming out with phase 1, phase 2, phase 3 clinical trial data in the next few months. So you understand what they're talking about and how they got those figures, and the limitations actually of those figures as well, so that you can understand how to not overinterpret them, which I think is a very important thing when you're liaising with patients who are going to ask you all these different sorts of questions. Next slide, please. Okay, part of my job is to really keep abreast of all these developments with different viruses, different vaccines, treatments, and diagnostics. And SARS-CoV is not any different. A lot of these vaccines have been produced in different ways for different viruses over many years, and you're very familiar with some of them, like hepatitis B vaccine, hepatitis A, rotavirus, MMR, VZV chickenpox, influenza, of course, and some of the travel vaccines like yellow fever, for example. So you're also familiar with the types of vaccines that are available. Now, with the COVID-19, the acceleration of the vaccine development has been quite extraordinary, and some of the technology is new. Now, it's new to human vaccines, but in fact, the mRNA vaccines that we'll talk about first have been around for a long time in animal vaccines and in anti-cancer vaccines. And that also includes the adenovirus as well. So in a sense, the technology is not new, but the way it's been applied at such a breakneck speed to the SARS coronavirus 2 pandemic has been quite amazing. So there's something familiar and something that are not familiar. I'm going to try and coalesce these together to give you an idea of what is going to be coming to you in the next few months. So if you look at this nice little pie chart, it shows that most of the vaccines we're familiar with are basically subunit or recombinant protein vaccines. So if you look at the 10 o'clock position, the protein subunit are things like hepatitis B, influenza, which you're very familiar with. The recombinant viral vectors, we don't really have one for current licensed NHS use, so that's new. We don't have a nucleic-based one. If you look at the 4 o'clock position, that's also new. And then the inactivated virus vaccines. In fact, we don't have those anymore. We used to have them. I think at the moment, I think it's only polio. The inactivated polio vaccine is the inactivated example there. We used to have an inactivated whole virus vaccine for influenza, but it was too reactogenic, too many adverse effects. So we removed that and replaced it with a subunit protein type vaccine. And the virus-like particles, that's really the HPV vaccine, the VLPs, and they work very well and produce a nice immune response with relatively few side effects. Now, if you look at the left-hand side, I've just listed the front runners in this COVID-19 vaccine effort race, although it's not supposed to be a race, but it really is a race. So if you look at the Moderna mRNA vaccine top left, you just heard about that last week. These are loading S protein, the binding protein for the SARS coronavirus 2. Pfizer-BioNTech, you've heard about that for the last week or so. And then earlier, the Russian Sputnik 5, the adenovirus vectored vaccine, again coding for S protein. And that's what we call a heterologous vaccine with adenovirus 5 and 26. We'll talk about that more later on. Oxford AstraZeneca, as you know, with the chimpanzee adenovirus vector vaccine, and then the one that hasn't had a lot of press recently, the Johnson & Johnson adenovirus type 26 vector vaccine as well. And then below that, we have more of the traditional ones. GSK Sanofi combined forces to give you a similar protein subunit vaccine that we use for flu, this time targeting the S protein again of COVID, of the SARS coronavirus 2. And then again, Novavax, again, producing a subunit vaccine. Additional is the whole inactivated vaccine from Chinese Sinovac, which we're not getting here, but we are getting the Valneva whole inactivated vaccine sometime next year. Clinical trials, phase 1, phase 2 may start at the end of December this year, so they're a bit slower. Next slide, please, Abbas. So I've listed all the vaccines here that the UK has actually paid for already. Okay, so they paid for these vaccines. Moderna was a last-minute scramble last week when those results came out, which was quite amazing. They scrambled and got 5 million doses, but of course at 2 doses per person, that's only 2.5 million people who would probably benefit from that. Then the Pfizer-BioNTech's 40 million doses, again 2 doses per person, that's 20 million people get that vaccine. And then the AstraZeneca Oxford vaccine, 100 million doses. So there's a phase 3. Novavax starting phase 3 now, this week, I think, recruiting 60 million doses. So again, 30 million people might benefit from that. And then the Janssen vaccine, 30 million doses, starting phase 3 trials now as well. And then GSK Sanofi subunit vaccine, 60 million doses. 30 million people, not really started phase 3 yet, and then Valneva really quite far behind. In fact, that factory is in Livingston, I think, and they're going to start phase 1 clinical trials at the end of this year. So in total, if you look at that, you've got about, what, you've got 20 million, 50 million, 30 million, 15 million, 30 million. You got more than the vaccine for everybody. One of the articles in the press suggests you got 5.2 vaccines for every person in the UK. Well, the thing is, you know from vaccinating people against hepatitis B that some people don't respond to that particular type of vaccine. So unlike COVID-19, there's only one hepatitis B vaccine really. In fact, there are a couple of mRNA pre-S vaccines available for hepatitis B, haven't been licensed yet. So with COVID-19, you have a lot more choice. And if you don't respond to one, maybe you don't respond to the mRNA vaccine, you might respond to, say, a adenovirus vector vaccine or a whole inactivated vaccine or a subunit protein vaccine. So you have some choice if you fail to respond to one of them. Next slide, please. So this is a bit more of a graphic to show how these vaccines work. So let's take this slowly because it's a bit of a busy slide. So if you look at number 1 at the top, okay, number 1 at the top is protein-based. That's the Novavax GSK Sanofi. This is the flu vaccine. Okay, this is basically the flu subunit vaccine, surface antigen vaccine that you give routinely. Okay, so this is not new technology. This is well tested and tried. We're just adapting this for the S protein for the COVID-19 vaccine. Number 2 is the viral vector vaccine. It's the adenovirus vector vaccine. They use adenovirus because it's quite a big virus. You can cut out some genes of adenovirus and replace them with the target protein, the S protein that you want to actually create an immune response to. This is not something we've actually been using in humans under the normal NHS immunization schedule. So this is being new, but it's actually been well tried and tested with other vaccines that have been developed for emerging infections and also anti-cancer vaccines as well. And if you look at the number 3, it's really the mRNA vaccine, which is really new to human licensed vaccine. It has emerging virus vaccines that not come to license yet. So this COVID-19 mRNA vaccine has overtaken those existing mRNA anti-cancer and other viral targets to come to the forefront ahead of the rest, actually, so fast, because mRNA vaccines are actually very easy to make, very easy to produce in large quantity, and they don't have the kind of additional practical issues with adjuvants, etc., that we see with the more traditional vaccines. Next slide, please. So this is a very nice review in Nature. If you can get hold of this, it really lists all the different vaccines, all the different strategies. It's a very nice review. It's a bit outdated now. It basically lists the strategy on the left. So I put the current vaccines that we have for COVID underneath that. So you can see Russian Sputnik 5 for Janssen and Russian Sputnik 5 for the adenovirus 26 non-replicating adenovirus vector vaccine carrying the S protein gene. You can read the pros and cons as you go across. Mostly it's IM parenteral injections, normally 2 doses, and they have slightly different immunogenicities, as you can see on the right. And then the last one is the chimpanzee adenovirus vector vaccine by AstraZeneca and Oxford. So again, these will be IM injections. You're very used to these, deltoid IM injections. You need 2 doses, usually between 2 and 4 weeks apart, which adds to the logistic problem. Next slide, please. And then the mRNA vaccines on the top there with Pfizer, BioNTech, Moderna, and they seem to be responding very well in the phase 2 and 3 trials so far. We don't know how often you'll have to repeat them because the vaccine induced immunity may not last. We don't know how long it's going to last actually for any of these vaccines, but we have some better idea with the subunit and inactivated whole virus vaccines that we're more familiar with. And we'll see how long these mRNA vaccines actually induce immunity for. We haven't got any DNA-based vaccines for COVID-19 as far as I can tell. And then the live attenuated, I think there's some actually being developed, but I haven't looked into those too much. But this review actually lists some of the alternatives here. I've just listed the ones that are most close to the UK vaccine program in this talk. Next slide, please. So this is how the mRNA vaccines work. So let's look at Moderna and replicating mRNA vaccines. Now, what does that mean? It means that the mRNA doesn't replicate, it doesn't amplify inside the body once you inject them. The virus gets injected, gets picked up by dendritic cells that then process the mRNA and translates the virus protein, expresses it on the cell surface, and induces that immune response that you can see on the right there with the B and T cell immune response. Now, the self-amplifying or self-replicating mRNA vaccines actually being developed by my colleagues in Singapore, by En Yong Oi, the professor there. And this is a self-replicating vaccine. So what that means is that, in fact, they could probably get away with just 1 dose and then not have to go back and give it a second dose. And in fact, it's a bit like MMR. MMR, you give 1 dose at 12 to 15 months, and then you don't have to give another dose till preschool, 4 to 5 years of age. So it's almost like a single shot, fire and forget, a Singapore vaccine. And the data's not out yet. They're looking at early next year to start clinical trials. And I'm very curious to see whether that actually does reduce to 1 shot. Because if it's 1 shot, it's much more convenient for the GPs and the patients. And it may be, it may be cheaper. The Pfizer, BioNTech and Moderna vaccines are likely to retail around $30 to $40 per shot. Well, per treatment, per vaccination course. So that includes both shots, actually. The Arcturus Singapore, I'm not sure how much that's going to cost, but it's half the amount of vaccine that you have to give. The Oxford AstraZeneca vaccine is much cheaper. It's about $3 to $4 equivalent. It's like 1/10 the cost of the mRNA vaccines, and that's something we should just keep an eye on. And the storage is different. The storage is 70, for Moderna is like -20. I'm not sure about the Arcturus in Singapore, but they may actually reformulate it to be, 2 to 8 degrees. And the Oxford AstraZeneca is probably going to be around 2 to 8 degrees, 2 to 8 degrees refrigerated temperature as well, which will be much easier for GPs to handle. Next slide, please. So again, just focusing on the mRNA vaccines, because this is quite unfamiliar to a lot of you. So again, I've just listed the vaccines that will be relevant here on the left-hand side. So the mRNA vaccine is really a messenger RNA vaccine. It carries the code for the gene. It's injected in liquid nanoparticles into your deltoid muscle that are then taken up by your dendritic cells. And then your dendritic cells will actually translate that mRNA directly into S protein from the cytoplasm of the cell. The actual mRNA is not infectious. It can't revert to wild type like we used to see in live polio. Although it says risky because it's new, it's actually not new technology. And there are a couple of other vaccines, mRNA vaccines, including an HIV, a few HIV mRNA-based vaccines in development, as well as for Ebola, for example, and Zika virus, and even rabies. Okay, and the mRNA vaccine, unlike say flu vaccine, won't have to be subcultured to amplify in eggs. So you might get people who are allergic to eggs not able to take the flu vaccine. We know about that. The mRNA vaccine won't have any of those issues in the amplifying system. Next, please. Now, this is a tricky issue. So what do you mean by vaccine efficacy? So does that mean that if I get vaccinated and someone coughs in my face with SARS coronavirus 2, I will be completely protected? We're not sure. Okay, so there's some talk about human challenge trials, which will be very interesting, looking at otherwise healthy volunteers for that. And we don't know what they'll find. They might find that, oh, I'm completely protected, so I don't even get infection, or I might get infection but no disease. So if I inhale the virus after being vaccinated, and you swab me, say 5 to 10 days later after the incubation period, you might pick up virus, but I may not be sick. Okay, and we don't know how infectious that virus will be. So that's a tricky question. And then, of course, it may not work at all. So you get antibodies that you can measure, but you still get infection, you still get disease, and you still get sick. And the reinfection cases that have been published recently, patients do get sick again despite having antibodies and being exposed and infected by the virus a second time. So we don't know the answers to these questions. And the efficacy studies, basically efficacy is really what you're going to see in a few minutes, looking at antibody T-cell responses in vitro in the lab from volunteers that they sample, post-vaccination on a periodic basis and measure these antibody and T-cell responses in the lab. The effectiveness is really what you see in real life. Okay, so some of those effectiveness or efficacy studies reported last week, when they claimed 95%, 94% efficacy, it was really from a phase 3 clinical trial exposure in the community to the virus. And looking at those who were vaccinated, only 5 got infected versus 90 who got infected when they didn't have the vaccine. Assuming that they all get exposed equally in the community in both arms of the trial. Okay, so that is a fairly big assumption, which may not be unreasonable when you've got widespread community spread ongoing. Next slide, please. So here's a calculation. So how did they get this 94%? And it's basically a relative risk calculation. I'm not a statistician, but I've got some good friends who have, and they help me with this slide. And they're online calculators. You actually estimate this. So if you look at some of you will be better at stats than me. But if you look at your vaccinated plus COVID positive exposed. You can see this is 5. I think this is from the Moderna vaccine numbers. And then the ones who actually were not vaccinated and got infected are 90. And if you assume a total population of 30,000 in the trial arm, 15,000 the vaccine arm versus 15,000 the placebo arm, then you just subtract those numbers from those totals. And then you do your 2x2 table and you work at the relative risk, and the formula is shown on the bottom right there. So if the relative risk is 0.0556, then your protective efficacy is 1 minus that, which comes to 94. Okay, so that's, I think that's the Pfizer figure. So that's where you see this coming from. Okay, so you can do this yourself when you see figures coming out in the press later on. Next slide, please. Okay, so the prime boost effect. So in fact, if you've not heard this term before, you've been doing this for most of your working life. When you vaccinate somebody with hepatitis B, your hepatitis B vaccination program is 0, 1, and 6 months typically for most adults. The accelerated version of that for hepatitis B or born to Hep B positive mothers is 0, 1, 2, and 12. Okay, so what you're doing is you're actually priming their immune system with the first vaccine dose, that's the prime, and then you're boosting it with the second one. So the boost might be 0 and 1, or 0, 1, and 6. So you basically priming it once and boosting twice. And the babies who get vaccinated are having it boosted again at 12 months. Now, in fact, if you test them after the first 2 doses, you might already have an anti-HBs response, you might have an antibody response. You don't need to boost them anymore. But the thing is, it's much easier to give the boost than to keep testing them, because you test them, they have to come back for the result, you have to send it to the lab. So we tend to just give the vaccine without checking in most cases that the vaccines worked. And you've seen the PhD guidance, which says that if you've had 2 MMRs, if you've had 2 you're immune. Okay, so that assumption is really based on some of the trials done where you might induce an immune response in like over 90% of people with the first prime dose, and then you catch another 90%, so that's 99 of the remaining 10% with the second dose. So if you think about the type of vaccines you use, most of the time you use this homologous prime boost regimen where you use the same vaccine twice. And you see this with the 6-in-1 vaccine, as well as polio and HBV, sorry. If you then use a different carrier, different formulation, same antigen, different formulation, with a prime boost, a heterologous response, why would you do that? And the Russian vaccine, the Sputnik 5, does this because the problem with the adenovirus vector vaccines is that your immune system will react as vector. And in fact, if you use an adenovirus 5 vector on its own, you probably have antibodies to that, because that's not an uncommon adenovirus vector. And it's been around, in the community, causing respiratory infections or sometimes eye disease, for decades. And you may already have antibodies to that. So when you get injected with that particular virus vector vaccine, you might neutralize the vector and the vaccine before it causes induces any immune response to COVID. Okay. So what the Russians have done, they use a prime, which is adenovirus 26, which is much rarer. They prime it with adenovirus 26, and then they boost it with adenovirus 5. Okay. The Oxford team used a chimpanzee adenovirus vector, which most of us have never seen before. So that won't have any neutralizing antibodies when you inject that the first time. And the Janssen just used adenovirus 26 and hope that most of you will not have antibodies to adenovirus. Now, of course, if you need repeated, you will remember, your body will remember, hey, I had this last year, and now I've got antibodies to adenovirus 5 and 26 and the chimpanzee vaccine. Repeated boosters the following year, if this has to be a seasonal annual thing like flu, those vaccines may not work so well the following year because you've got antibodies to those vectors, and either they change the vector or you use a different vaccine. So this is something that they have to be careful about, because the effectiveness of that vaccine may wane after just 1 dose. We don't know. Next virus, please. Sorry, next slide, please. So now we're just going to go through the different vaccines. So this is Moderna. We've gone through the mRNA process. This is a nice, more beautiful slide than the earlier one. Next slide, please. Now, I've just shown the side effects first. So if you look at the left, I've summarized there's not many people, okay? So there's 2 IM doses, 28 days apart, dosed at 2,500 and 250 micrograms, just 15 in each group. Age range is 18 to 55, and follow-up is 7 and 14 days post-vax, and longer 57, 119, and 294 days post-vax as well. That's pretty good. Now, they found that, in fact, if you look at those adverse effects, the 250 causes too many adverse effects. So in fact, they've actually dropped that for the subsequent study, subsequent trial study, and they're focusing on the 100 microgram dose that you can see there in the middle of those bars. And basically, those bars show you the adverse effects going on the left-hand side. That's arthralgia, fatigue, fever, chills, headache, myalgia. So that's the systemic adverse. Lower down, you have local erythema, induration, and pain. And basically, yellow and orange is bad, blue is not so bad, and grey is better, is the best. Okay, and most of these adverse effects are worse after the second vaccine shot. Okay, I'm not sure whether they actually injected into the same side of the deltoid. It may not matter too much if it's 28 days later, but clearly the second shot is more immunogenic and it causes more adverse effects. I don't know how that compares to the other vaccines. This is very subjective. But they all kept pain diaries, and some of them used solicited adverse effect questionnaires. Now, if you look at the next slide, this is a very common type of slide you can see. If you look at the bottom axis, basically looks at the different dosing and the time post-vaccine dose. Okay, so there's 2,500 micrograms, 250 micrograms going left to right on the bottom axes for each of these slides. And then up the side, you have the reciprocal endpoint titer. Now, what this measures is just how much antibody you produce by whatever method they decide to use, ELISA, EIA, microneutralization antibodies. So PRNT is plaque reduction neutralization titer. PVNA is a neutralizing antibody assay. And then the top two are basically different EIAs. One's receptor binding domain, which is a subunit of the S protein, and the top left is just a whole S protein. Okay, so what you're looking for is basically a rise in the curve over time. Okay, and if the curve rises higher over time, that's a good response. Not a good response. Okay, if you look at, say, the top left to start with, you find that the 100 microliter, so the 25 microgram dose induces a peak response about day 36 that is slightly lower than the 100 microgram dose, and similarly that's slightly lower than the 250 microgram dose. Okay, but not by much. But if you look at the left-hand axis, the vertical axis is actually a logarithmic axis. So a small difference could actually be a big difference because of that axis. Okay. Now, of course, the trade-off is the adverse effect. So if you give a massive vaccine dose, you'll have a massive immune response, which is great, but your patients may become very sick because of that. So they have to figure out what's the optimal vaccine dose to give that doesn't put your patients off having the vaccine in the first place. Okay, so a lot of these types of slides going forwards now. Okay, and they've also looked at the convalescent antibody levels. If you look at the top left, top right, and bottom left slides, pictures there, the very far right bar is the convalescent antibody level of those who've actually had COVID-19. And you can see that the spread of the antibody titers neutralization assay or EIAs is actually fairly similar to the spectrum of titers you see from the vaccine-induced antibody, and covers the whole range of responses actually from, early day 15 up to day 43. Okay, so they're in the right ballpark to induce a response that's similar to the convalescent antibody from natural infection. Next slide, Abbas. Okay, so Moderna have claimed 95% effectiveness for this vaccine, which is based on just, I think, less than 10 people who have got infected versus about 90% who got infected, and the less than 10 is the vaccinated group, and then the greater than 90 or so is the non-vaccinated group, and that's based on that calculation that I showed you earlier. Next slide, please. Okay, so the Moderna vaccine can be stored at -20 and for long term, but can be stored in the fridge for, I think, up to a month, which is quite useful. The Pfizer-BioNTech, I think, has been rushed out without giving it to be stable at a warmer temperature. But again, the results are very good. Now, this paper actually looks at two different versions of the BioNTech vaccine. One is RBD only, that's receptor binding domain, which you saw in the previous slide. And one is the whole S spike protein antigen coded by the mRNA. And again, at 4 different doses, 10, 20, 30, 100 microgram doses versus placebo. Again, just 15 participants in each group, so very small numbers. But this one across a higher range of age groups. Now, they looked at which one to include in the vaccine, either the B1, RBD only, or the B2, the whole S protein coding mRNA. And surprisingly, they found very clearly that the whole S protein coding mRNA induced relatively than the B1 RBD, which is a smaller protein, a smaller antigen essentially. It's a subunit of the S protein. So if you look at the red boxes show the B2 dose 1 and dose 2 adverse effects versus the graph above those red boxes, which is the B1 first dose, second dose adverse effects. And you can see there's a big difference there. I'm surprised there's such a difference, but clearly they favor the B2 vaccine option because it has fewer side effects with relatively similar immune responses. Next slide, please. Okay, so this I don't actually have any data on the neutralization response from Pfizer, but this again is from, these are looking at the older age groups. So I think this is 65 to 85 years of age. Now, there is some data. They said that it shows equal efficacy across all age groups, but I haven't actually seen that data because unlike the other vaccines, Moderna has a paper, AstraZeneca, Oxford has a paper, Russian Sputnik 5 has a paper, but the Pfizer is really all press releases. So I haven't got this kind of neutralizing antibody EIA data from the Pfizer-BioNTech mRNA vaccine, just the endpoint calculations. But again, this just shows that the B2 protein that they code for the mRNA vaccine is induces fewer, less severe adverse effects across all age groups. So this is like a kind of preamble to the vaccine they actually use. Next slide, please. Okay, so again, they claim 90% effectiveness. Now you can see how they got the calculation. I think it's like 8 people who got vaccinated who got infected with COVID-19 versus about 87 who didn't get the vaccine who did get COVID-19. So it's very small numbers. If you've got the same percentage efficacy, where they involved 1,000 to 10,000 participants, because they've got 40,000, would that be much more convincing to me? And as the trial progresses, you might find that some of those numbers will change over time, and the efficacy may actually not be as high as 90%. That will not be that surprising, but we'll see. Okay, so next slide, please. So here are some useful questions from from the press, and I think some of these are: can the vaccine prevent severe disease? Well, okay, the vaccine should prevent any disease, hopefully. If you look at MMR and chickenpox, and well, let's look at MMR first and chickenpox, where the vaccine actually prevents wild-type virus causing disease, but sometimes the vaccine itself, the virus in the vaccine, these are live vaccines, can cause some mild disease. Okay, so we don't know how much disease these vaccines will actually prevent. It's interesting that if you look at the reinfection of by COVID-19 in the cases that have been reported, some of them actually show that even pre-existing naturally induced antibody, they don't prevent moderate to severe disease either. So I think there's lots of individual variation in this. Generally for respiratory viruses, RSV, viruses, you can get reinfected several times a year. Now, it may not be with the same serotype, because there are over 100 plus serotypes of rhinovirus and the 4 different circulating coronaviruses, but you can certainly get infected, year on year between, even with the same serotype, because the antibodies don't last, the immune response don't last that long for respiratory viruses, and the various reasons for that. Does the vaccine prevent transmission? Well, again, it depends how you respond to that vaccine. If you respond in a very strong way with massive neutralizing antibodies and massive T-cell responses, you may not even get infected. If you don't get infected, you can't transmit because there's no virus circulating and shed by you from the oropharynx. So again, it depends on how well you respond to the vaccine. Now, we know that flu, the vaccine isn't 100%. So people can get vaccinated and can get flu, and then they can actually have milder infection or flu vaccination despite exposure to flu. So I think there's a sliding scale of responses to all of these questions based on the vaccine and individual immune responses. Does the vaccine work with older people and children? Children, we're not sure yet, because I don't think we've got that much data for children. But for the adults, the older adults, there is some data now from Pfizer that shows that even adults of up to 85 can actually respond very well to the vaccine, which I find quite surprising, and it's great if it's true, but normally older people don't respond very well because of immunosenescence kicking in, and none of the other vaccines do very well in elderly, particularly flu, where you need double adjuvantive vaccines to help them produce an immune response. How safe is the vaccine? Well, it depends what you mean by safe. I mean, inevitably, somebody will come down with Guillain-Barré syndrome, or some severe adverse effect from some of these vaccines. If you vaccinate 1 million to 60 million people, even a 1 in a million complication like Guillain-Barré, you may get some cases. And in fact, you can get Guillain-Barré even with natural infection from flu. So that's not that surprising. Why doesn't it work for 10% of people? I think it's too early to say. You've only had results back for a couple of weeks for probably less than a couple of hundred people. So we know that there's a baseline failure rate for all vaccines of about 1 to 5%. For various reasons, some people just don't respond, they're just non-responders. Sometimes it could be practical things like failure of cold chain. It's very difficult to pin that low 1 to 5% failure rate down to a specific reason that applies to everybody. I think a lot of you will have had healthcare workers like midwives who've had one course of HPV, 0, 1, and 6, no response. Then another 1, 0, 1, and 6, no response a year later. I've had one midwife of Hep B vaccinations a few years apart, and she finally responded to the last one. So I started the 4th round of vaccination. This one I was at UCL, and she actually had an anti-HBs response. Now the other thing is how you measure the response. So a lot of GPs they may not even test for anti-surface response, And if they do, they might take the sample maybe too long after the vaccine. So some people take the vaccine response sample, say, 6 to 12 months later, and then they're negative. There's no anti-surface. But I said, well, you can't say there's no response because you're supposed to take it within the first 2 months after the last dose of vaccine to see that response. So this type of surveillance hasn't been really thrashed out for these COVID-19 vaccines. How do you define a response? And when should you take a sample to actually define that response? How long does protection last? Well, we don't know. We don't know. So and look at all the studies done on natural infection for the past 6 to 8 months. We think we have neutralizing antibodies induced by natural infection for the 3 to 4 months following natural infection. And then after that, your anamnestic response, B and T cell response might kick in, and you might have long-term B and T cell memory responses that are prime responses. Even though you lose your antibody, if you see COVID-19 again, you might get a rapid response, which is like other viruses, and you might be able to neutralize the virus before it even causes infection in you. But again, we don't know. We don't know. That needs a longitudinal follow-up, probably in phase 4, once the vaccine's licensed. Next slide, please.
Julian, just a quick point. So your voice keeps cutting sort of every minute or so. So someone suggested maybe if you turn your video off, it might strengthen your audio connection.
Right, hang on. Okay. Okay, sorry about that. I think my Wi-Fi's not working. So, okay, let's try this. I think it's probably buffering a bit. Okay, so we had an update last week of Pfizer, the Pfizer vaccine saying 95% effective, including I think the elderly as well. And this one was very well balanced for ethnicity. Okay, so the Oxford AstraZeneca vaccine didn't include any Asian BAME population at all. I think like less than 1%. It's really a all-white vaccine trial for the Oxford AstraZeneca vaccine, whereas Pfizer and I think Moderna to some extent really tried to include the BAME population. And that, I think that's a fairly important issue because we know the BAME population is suffering more from COVID-19, at least in the Americas, than the white population. I think that these extra figures, 170, 162 in placebo, 8 in the vaccine group, just encourages us to believe that it actually is very effective. But if I can see like 10,000 people involved in those figures, I'd be more reassured. And this also highlights the fact you need to transport and store this virus vaccine in dry ice because it's -70 before defrosting it and giving it all within 5 days. I don't know whether you're going to get those boxes in your practice. I suspect not, because dry ice can be quite dangerous. Dry ice has to be in a leaky box, okay, because it sublimes, and if you keep it in a sealed box, the box will explode. So these boxes should be not taped down and airtight, others are going to have a little bomb in your surgery. I suspect they won't give you these boxes in your surgery. You'll get it from a central depot, which we'll hear about later on. Next slide, please. Okay, so I'm just going to go through this AstraZeneca vaccine, and I've highlighted here that basically very few BAME participants in the trial, phase 2-3 trial now, and my impression is that there's more adverse effect with this adenovirus vector vaccine than with the mRNA vaccines. There's no head-to-head on this, but it's just my impression. If you look at the data, there's a lot more higher frequency of mild, moderate, severe adverse effects. And I think this may be an issue with this vaccine. Even though the storage is nice, the price is nice, I think people will get more adverse effects from this vaccine because the adenovirus vector is quite reactogenic. Next slide, please. Okay, so the Oxford team have really gone over. I think they've gone overboard with all these this data presentation. I think they want to really show everything. but it gets a bit confusing. So if you look at the two sides of this slide, basically shows the adverse effects. On the top, you can see what they are. Okay, there's the local and systemic. Very small on my slide. So yeah, so you got pain, you got swelling. I think you've got let's have a look. It's very small here. Tenderness. Yep. So you can see that the histograms speak for themselves. So you got a lot more pain and tenderness on the groups. Slightly less in the older age groups. Like if you look across the rows there. And then on the right, you've got the systemic effects. Again, fairly prevalent across all age groups, more so in the young than the old. But it seems more reactogenic than the mRNA vaccine adverse effects. You can look at this more closely later on, but also don't forget that different people react in different ways and report things in different ways. And it's interesting that they use paracetamol. So they're aware that this vaccine is actually quite reactogenic. So they actually have an arm where they give paracetamol versus no paracetamol to see whether it actually helps reduce this. If you look at with paracetamol, without paracetamol, I don't know whether paracetamol makes a lot of difference, which means that for the first 5 to 7 days, your patients some of them will experience some adverse effects from this vaccine, which might put some of them off. The local pain and tenderness is not that unusual with flu in the deltoid, flu vaccine in the deltoid, you get this. But, again, it lasts 5 to 7 days, which is quite a long time. So I guess you'll see. Next slide. Okay, so again, the second dose seems to be less reactive adverse effects after the second dose in this particular cohort. Most were after the first dose, the priming dose. And again, you can see that pain and tenderness are the prominent adverse effects locally. And then you've got chills, fatigue, fever, and then headache, malaise, muscle ache. In the systemic effects, again, worse in the first dose. So in fact, if the first dose is so bad, they may not come back for the second dose. So I don't know how the patient's going to react to this. And these effects last for, again, 3 to 4, up to 5 days. So we'll see. Next slide, please. And again, these graphs that I went through earlier with you, there's a ELISA on the left, and that's the meningococcal vaccine. You see, it's very interesting. In the mRNA vaccines trials, they just use placebo, so saline versus the vaccine, mRNA. In the Oxford trial, they realized that the adenovirus vector vaccine is quite reactogenic. It causes quite a lot of adverse effects. So if they gave saline, they say in the paper, if they gave saline, the people would know that they haven't got the vaccine because the vaccine was expected to give adverse effects. So instead, they gave the meningococcal vaccine to kind of mask that adverse effect profile of the adenovirus vector vaccine. But in fact, the meningococcal vaccine didn't cause as much adverse effect as the COVID-19 vaccine, the adenovirus vector vaccine. So the team were aware, plus also they gave the paracetamol dosing as well. So the team are aware that this vaccine is quite reactogenic and has quite a lot of adverse effects compared to other vaccines, because they used a placebo, which actually was the meningococcal vaccine, to kind of try and hide some of that adverse effect. And also they gave the paracetamol option as well. So I think the team know this is quite a reactogenic vaccine, but they say that the adverse effects were tolerable in the papers, in the 2 papers they published. And again, if you look at the convalescent plasma here on the right, you can see that the immunogenicity of the vaccine is on par with natural infection. Okay, so if you look at the titles, you've got prime, which is meningococcal on the left, prime with the first dose of adenovirus vector vaccine, and then prime boost, which includes a second dose. And you can see the second dose does actually boost the antibody levels slightly. Again, this is a log scale on the left, so just be aware of that. That's in the antibody, and the convalescent plasma covers the whole spectrum of that. Next slide, please. And this is the T cell mediated response. So this is looking at your cell mediated response, and again, it shows it's less convincing for the prime and boost titers, looking at interferon ELISpot assay responses, but clearly they elicit a stronger immune response to the virus than the meningococcal vaccine, which makes sense. Okay. Although interestingly, the meningococcal response is actually not 0. Okay. Next slide. Okay, so this is the follow-up study to the first study that we just went through. And this time they used the lower of the vaccine. So they've chopped and changed a lot. They've moved around a bit. And they say that the low dose is because of the manufacturers that have created a low dose, and then there's a standard dose. And I'm just wondering whether the low dose is because they know that the higher doses give more adverse effects to this vaccine. So it's very interesting to see, how they respond to this. Again, I've just highlighted this phase 2/3 study follow-up is actually basically all white as well. Okay, there's hardly any Asian or black. There's no black. I think there's 1 black person and about 10 Asians. 13 Asians. Okay, so it's not a great representation of the BAME population, which is suffering the most actually from COVID-19, which is a bit of a pity. Oh, next slide. Can you hear me okay now? Is it better if I turn off the video?
Not that much better, but I think it is. I guess it is what it is, unfortunately. But
okay. So I think it might be my Wi-Fi here, which is having some problems tonight. So anyway, I'll say, I think I say most things a few times anyway. Here you can see the structure of the trial, and this is a really complicated structure. So you can see that they're looking at basically low and high dose, looking at prime and prime boost across different age groups. Okay, 18 to 55, 56 to 69, and greater than 70 and above. Okay, so this is a really complicated that the vaccines haven't got into. And in fact, one of them will have paracetamol, no paracetamol in it as well. That's the previous, I think that's a previous paper. So it's a very complicated design, and I just like to see more data coming out of this vaccine before I can really make up my mind about this. Next slide, please. And again, the injection site pain and tenderness are the main local adverse effects, and they're clearly the most predominant here across all age groups. Slightly less in the elderly, which might be due to the immunosenescence and less of an immune response to the vaccine, causing those adverse effects. And then next slide, please. You look at the systemic effects, adverse effects. Again, less prominent in the elderly, more prominent in the young, 55, which would be the bulk of your patients, I guess. And And I'm just wondering if the first dose is so reactogenic and so uncomfortable, will they come back for the second dose? We will see. You can keep me updated on that. And some of these will last, 5 to 7 days, which might be quite uncomfortable for some of the patients. And there's quite a lot of orange and red there. So they're not just mild adverse effects. So despite the price and the storage advantage of this adenovirus vector vaccine, the adverse effects after the first dose might be off-putting in a lot of patients, I suspect. Next slide. So again, you see these nice graphs. The Oxford team have very good at showing these graphs. So you're seeing the rise and titer measured by various different methods, neutralization, EIA, RB, as in the first paper, showing a nice rise up to day 40 something, and then plateauing off. So you can see a detectable response up to day 56, it looks like, so at least 2 months. Beyond that, we don't know how long the immune response will last. Next slide, please. And again, here's just looking at these T-cell responses, showing that, again, they rise and plateau around day 28 and last until 56. So I'm not worried about producing a B and T-cell response in the short term. Long term, we don't know. Same with the mRNA vaccines. Next slide, please. Okay, I just covered the Russian one quickly because it's out there. It's a heterologous prime boost response, and it's just and you can see that the design of the. Okay, so you got what they've included is actually a frozen vaccine version and a lyophilized vaccine version. So what does a lyophilized mean? It means that it comes in a powder, and then you just reconstitute it with water or sterile water injection, water for injections, and then you can store it at room temperature. You don't even need a fridge. Okay, and again, the relatively small numbers: 38 in one arm, 38 in the other, and then basically 9, 9, and 20, 9, 9, and 20. But this is much easier to follow, very straightforward. And in fact, the Russians and the Chinese have been developing vaccines for a long time. So they have a good history and a good track record of vaccine development. So I wouldn't doubt the vaccine quality for that reason. Both the Chinese and the Russians have actually started using vaccines in the general public before phase 3 clinical data was made available. Okay, that's low in the Western countries, but it doesn't mean the vaccine is necessarily bad. Okay, next slide, please. So again, this is really just looking at systemic effects, which again is very similar amongst these vaccines. So fever, headache, fatigue, myalgia, arthralgia, however you want to phrase it, and local pain, swelling, and fever. I don't get the impression that this is any worse than the Oxford vaccine. Of course, the numbers are very small, and they present it in a slightly different way. They don't look at the duration of the adverse effects like the Oxford vaccine team did, but I'm guessing it'll be within like 5 to 7 days as well. Next slide, please. And again, these nice graphs showing humoral responses, that's antibodies responses in the frozen version on the right, lyophilized version on the right, and then they have the prime boost adenovirus 26 plus adenovirus 5 on the far right. Okay, so they've looked at the adenovirus 26 first on its own, and then the adenovirus 5 on its own, and then the combined adenovirus 26 and then the 5, given I think it's 21 days apart. And you can see that if you look at the combined prime boost response on the far right, it does increase the antibody titers quite a bit more than the individual prime vaccines on the left. So you can see that quite easily visually. So having both vaccine shots is clearly more beneficial than just having one. And the difference is, again, this is it's actually a log 2 based vertical axis. It just doubles on the axis. But you can still see that the antibody response is higher if you have both the prime and the boost shots of the vaccine, which is not surprising. So I'm not sure what the I don't know what the spread of the efficacy is across age groups and BAME ethnic groups in this particular vaccine. And looking at the lyophilized versus frozen, and also the adenovirus 5 versus adenovirus 26 responses, really. Okay, next slide, please. And this is, again, looking at the T and B cell responses. So the last slide was the B cells response. Now this is the T cell response. Again, looking at the prime adeno 26, the prime adeno 5, and then the prime boost adeno 26 versus plus adeno 5 in the frozen versus the lyophilized version of the vaccine. The titers do rise over time for both the B and the T cell responses, which is encouraging. But again, we don't have much data on the age-related performance as well as the ethnic BAME population performance. I suspect most of these are white Caucasian participants in the Russian vaccine trials as well. I think that's it, isn't it? Next slide. Okay, and then this is looking at the neutralizing antibodies using a different assay. This is actually, the previous assays were like ELISAs. These are neutralizing antibodies with the prime adeno 26, prime adeno 5, and then the prime boost adeno 26 versus followed by adeno 5. And again, this different assay, neutralizing antibody assay, shows basically a slightly better response with the prime boost combination of adenovirus 26, adenovirus 5, which again is not that surprising. I can't really see much difference between the frozen versus the lyophilized version, so probably both are equally okay. The lyophilized version clearly doesn't need a fridge, need a freezer. You can store it at room temperature and transport it at room temperature, which is much easier and much cheaper and much safer. Okay, next slide, please. So this is just a summary slide. Basically, the factors that may decide which vaccine people stock and use is the usual, price, availabilities, delivery, storage, administration issues. The adverse effect profile, I think, will affect a lot of the uptake. If they have a choice of vaccines that perform equally well, then they'll probably choose the least painful vaccine. I mean, that's what I would that's what I would do. We don't know really about the degree of protection or the nature of protection, how much it will change over time, and whether we need to have repeated boosters each year. In terms of transmissibility, well, this is a bit of a red herring question, because if everyone gets vaccinated and the vaccine works well, even if you transmit the virus or you shed virus that can be transmitted, if you're vaccinated in a vaccinated population, this shouldn't be an issue. So that's a question that, again, is phase 4 surveillance to answer that. And the duration of protection, we don't know. We don't know what the nature and duration of that protection will be. And they may differ between different vaccine types and different individuals. Again, that's a phase 4 surveillance issue. So I hope that helps. I'm really sorry about the Wi-Fi. I don't know what's happening tonight. But if there are any questions, I'll try and answer them.
Brilliant. Thank you so much. The questions are coming in thick and fast. So yeah, you open up your video so we can see you, and then we can get you to answer some questions. So we've had quite a few. So if I start from one of the ones that came in quite early on, you mentioned about the fact that there are some vaccines that we will respond to, some vaccines that we won't. So how do you know you haven't responded to the vaccine other than obviously contracting the disease?
Oh, so we can do antibody testing. So this is the thing. The vaccines aren't given to those who don't have any antibodies. They're just going to be given out. So we don't know. We don't know whether you've had antibodies before, and when we test you after the vaccine, we're picking up those natural antibodies, or we're picking up the vaccine-induced antibodies. So we don't know that. But normally you can test for post-vaccine antibodies and see the response. And we can do that for Hep B, for chickenpox, for MMR. We can do that. It's not a problem. For even rabies, you can do that. So I don't know what the strategy might be. It might be just that you get vaccinated twice and that's it. Because we only do that for Hep B for healthcare workers to make sure they can go to the wards. We do that for medical students going elective so they can go to the wards. So, but for most people, you give MMR, you don't check the MMR response. And you don't check the vis-a-vis response, if you're in the US or other European countries where they give universal vis-a-vis vaccine. So you can check it with antibody testing, but ideally you want a pre-vaccine sample and a post-vaccine sample to compare.
Sure. Okay. And what about people who are pregnant or people who are suffering with cancer? I mean, how do we check the safety in those populations?
Yeah, well, that's the thing, that the clinical trial data is always limited, right? We don't vaccinate young children, we don't vaccinate babies, we don't vaccinate pregnant women, or those with cancer, or those with, other debilitating diseases. So the clinical trials are always going to be incomplete.
Yeah.
I mean, theoretically, the inactivated vaccines like flu, like Hep B, the protein subunit vaccines, so those vaccines can be given safely to pregnant women. So maybe the subunit vaccines can be given in the same way for COVID-19. So like I think it's the Novavax and the GSK Sanofi vaccines, you might have to give it in the same way.
Sure.
But we don't know.
Sure. So what about the risk of antibody disease enhancement? Has that been quantified? Because we know with previous coronavirus vaccines, MERS and SARS, they've had the same problem. And then we heard about the COVID-19 and things like Kawasaki's disease.
Yeah.
With the limited testing of the vaccine so far, has this been considered?
Yeah, so obviously I'm glad somebody listened to my earlier talk where I mentioned this as a concern. Yes. Okay, so these vaccine trials haven't looked at that, and they show pre-existing coronavirus antibodies. But there are other papers that have been published looking at this, and there is a good one which I can forward to people who are interested that suggests that the risk is low, but it's not zero. Okay, so the question then becomes: if you immunize against COVID-19, will the seasonal coronaviruses cause more severe disease if they interact with those COVID-19 induced antibodies from the vaccine? Okay, and I suspect if you're going to immunize, 7 billion people or 30 to 60 million people in the UK, you might see some cases of enhanced disease from the seasonal coronaviruses because of that COVID-19 vaccine induced antibody. I suspect you may see some. I don't know how much. In fact, I don't even know how they're going to tell, because unless you have a pre-vaccine serum that you can test and profile for the coronavirus antibodies to see what the COVID-19 vaccine has done to that, and then a post-coronavirus infection antibody profile to see what's happening with that, and a viral load for the coronavirus antibody, the infected coronavirus as well. So that could be quite tricky to follow up, but someone's going to do it. I'm sure someone's going to do it.
Sure. Okay, and I guess so. The question that's in my mind, I'm sure it's in a lot of minds, is that if you were listening to this slide set yourself, which vaccine would you personally choose, or would you choose not to have it at all?
I think I'd go for one of the mRNA vaccines. Actually, having looked at all this data, and I think mRNA vaccines look fairly safe. They're less reactogenic, and the mRNA. There's no real live vaccine component there. Okay, it's not going to be, so that's probably why it's not so reactogenic. So there's no real virus protein there. It's a bit of a liquid nanoparticle that goes into the dendritic cell. I don't know how effective those mRNA vaccines will be when you boost them every year. It may be the same, unlike the adenovirus vector vaccines that might actually start to lose efficacy because you develop antibodies to the vector. So the mRNA vaccine might actually be the one I choose.
Okay, so that would be the Pfizer, Moderna.
Yeah, Pfizer or Moderna for now.
Sure. Okay. And in terms of the dose, how do you decide what sort of dose you need to be able to develop the antibodies? Or is it just like a random number
for you? So the clinical trials have done that for you. They will pick a dose, and that will be the dose that they register and license. So normally they pick a high dose and a low dose, and usually it's the one in the middle that is most optimized for immunogenicity and reduced adverse effects. So in fact, you won't have to make that decision. That people, if they have one shot and it's so uncomfortable, they may not come back for another one. And then of those people who have a single shot, well, you saw from the data, a single prime shot still produces a response, but just not as much as a prime boosted response.
Sure. So in terms of the licensing, how are the MHRA pushing this through so quickly? How can they be approved using such small numbers? Is it just the emergency COVID law?
Well, Pfizer said they're going to have another 3 weeks of data, so they're going to submit to the MHRA. Well, I think FDA first at the end of November. Okay, and same with Moderna. They're going to wait till they have enough data. Now, how much is enough data? I don't know. I don't know how they decide that. I mean, they've got 30,000, 40,000 people in trials around the world. USA, Brazil, UK, and South Africa, I think. But how many participants from each group would they need to see data for to make that decision? I don't know how they're going to decide. I mean, more the better, right? But South Africans have a certain genetics, Brazilians have a certain genetics, USA, UK have different genetics. How are they going to look at that? And how many will be BAME population related? Pfizer said lots of BAME population participants is good. So I trust the Pfizer vaccine for ethnic minorities. The Oxford vaccine has hardly any BAME population-related participants, so we don't know how that will work in Asians or Black Africans. We don't know.
Would you tell patients that the vaccine has been rushed through? Would you avoid even having that discussion?
Well, the patients watch the news. That they can tell from the news what's going on. You don't have to. I don't think you have to say anything. Yeah. If they ask you, Dr. Tejani, do you think this vaccine is safe? Do you think it's been rushed through the safety process, the analysis? They're all saying that they haven't been rushed through. They're saying what they do is they're running phase 1, 2, and 3 in parallel. But the thing is, the safety data only comes from that data. The safety profile only comes from that data. So if that data is not complete yet, you can't say for sure how safe it is. So on the radio, I was asked this the other day, and they said, Dr. Tang, would you give this to your family? I said, well, I have to see the data first. He said, yeah, well, you know what I mean. Would you want to see would you give it to your family? I said, I have to see the safety data first, because we only have 100 participants, to base that efficacy and that adverse effect profile on. If I can see 1,000 patients or 10,000 patients worth of data, then I can make a decision. And that's not being annoying. It's just really, that's what I want to see.
Sure. And what if you've had COVID before? Would you still suggest having the vaccine?
So this is a tricky question. So most of the time, you don't check for it, but I know I've got antibodies, and a lot of my healthcare worker colleagues have antibodies. Those antibodies, I think they're protective to some extent. Okay, this is your natural response. I think they're protected to some extent. I've had mine rechecked, and in fact, I've got even more antibody now than I had 6 months ago. Okay. So I've probably been boosted in the community, or is assay variation. But either way, they haven't dropped, because you'd expect them to drop after 6 months. So I probably had some boosting from exposure in the community. Would I take the vaccine, knowing that I've got antibodies and lots of them? I wouldn't rush to take the vaccine, because I don't think that I need to take the vaccine because I've got those antibodies and probably prime B and T cell responses. Okay, so I think that people say this because they want to be cautious. But look, we've evolved with viruses over 2 million years. Okay, your immune response is going to react in a way to protect you to the virus, and respiratory viruses are always around. So I think that people may be underestimating how much immunity you have after natural infection. Would I take it next year when my COVID-19 antibodies have waned? Well, again, I need to see the safety data. Okay. Yeah. And we don't know what the interaction is of the antibodies that you have already from natural infection with these vaccines. Okay, so the antibodies are made to the S protein and to different proteins of the virus. They may neutralize those subunit protein vaccines like the GSK, Sanofi, and the Janssen, I think it's the Janssen, okay, or the Novavax. They may not have much effect on the mRNA vaccines, okay, because you don't make you can make antibodies to mRNA, but they may not have the protein antibodies, okay, because you make antibodies to all the different components. Okay, so you're going to have to see. You have to get some volunteers to do longitudinal follow-up, and we've had the antibody and given different vaccines and see what the vaccine response is in those people with those pre-existing antibodies. And someone's going to do that.
Yeah. There's a question about the likelihood of which of the vaccines has the least likelihood to give Guillain-Barré as its side effect. And I guess you've sort of answered that with the mRNA versus adenoviruses. So I'm guessing it's the mRNA virus vaccines that have the least likelihood of developing Guillain-Barré.
So the thing is, we have no experience with the mRNA vaccines. Okay, although there are several in phase 1, phase 2, no one's really brought one to license. If you look at natural infection and vaccine-induced Guillain-Barré, probably a response, a abnormal or aberrant host immune response to the virus protein, either from natural virus or the vaccine virus antigen. Okay, so I'm guessing that the mRNA is probably the least reactogenic in that respect, and maybe the safest from that kind of point of view. But they may have other adverse effects that we don't know about yet. So that's the only thing we don't know.
Sure. And is the mRNA synthetic? Are you lab manufactured? Is it porcine?
Oh no, it's synthetic. It's synthetic. So they probably they're probably. Well, in fact, I got a slide on this, but I didn't show it because it's a bit involved. But you can actually make your own RNA. You can actually get a synthesizer and make your own RNA, and make your own DNA. You can actually create it like Lego.
Oh, wow.
Yeah, so you can actually synthesize the whole thing. So I'm not sure how they made that mRNA, but maybe they just created it from scratch.
And I've got a question here about which vaccine gives the highest measured immune response.
So it's hard to tell because they use their own assays for each of these trials. So you have to go back and look at their, try and get equivalence between those different assays. I mean, some of them look at S and RBD, but the way you create those ELISAs and EIAs is a bit different, and the way you read them might be a bit different. So although they're called spike protein and receptor binding domain protein ELISAs or EIAs, they may actually have their own way of making that assay and running that assay in their own lab, and it may not be equivalent to another lab. So it's a bit hard to say.
Sure. Another question. So there's a big anti-vaccination group and it's growing fast. What can we do to convince the conspiracy theorists? And I'll give you an example. This morning, I had a chap come to my house to fix my roof. And he said, come on, Doc, you and I know each other. We go way back. He's been a few times to fix my roof. And he said, come on, is this coronavirus actually real? And I said, of course it is. He said, no, it's all Bill Gates. You know, he doesn't want any children. He's trying to cull the population. He's going to put nanoparticles in the vaccine. And I just said, yeah, just go and fix my roof. We'll have this discussion another time. But yeah, what can we do to convince these people, particularly given that the safety data isn't there?
The safety data is
limited.
It's limited, yeah, but I think it'll grow. But that's not why they're rejecting the vaccine, though, is it? I think they're rejecting the vaccine because of other things, like they're worried about you implanting biochips to control the population, or implanting some kind of, hidden symbolize you and all your family. I mean, that's the kind of thing that I've been hearing. It's not because of the mild safety concerns of the vaccine. Absolutely. I think it's a conceptual thing that is preventing them from taking the vaccine.
Sure. But what can we do to convince these people? I guess that maybe there might not be anything we can do to convince such people.
Everybody has a right to refuse the vaccine. Okay. They don't have to give a reason. They can just refuse not to have it. Is this within their civil liberties? It may well be. Okay, so if it's within the civil liberties, I don't try and convince a friend of mine that red is his favorite color. I say, well, why is it red? It should be blue. I don't think that it's worth fighting them for this. Now, if they go around trying to convince other people not to take the vaccine, that's a different thing. Okay, so what you decide to do on your own is one thing. What you try and convince others to do against their will is a different thing. But I think the best thing is to basically show this communication, to show people that the vaccine is safe and that it works and that you're better taking it than not. Yeah. And that's all we can do.
Absolutely. Okay. And just going back to the Novavax, GSK, Sanofi vaccines, which are in development, could they have potential benefits over the mRNA vaccines?
Well, the main those subunit protein vaccines or whole inactivated vaccine protein is just something that we're familiar with. And GSK, Sanofi, who produced the flu vaccine, they're geared up to produce subunit protein vaccines. So the whole machinery is geared around that. They just substitute a flu antigen for a COVID-19 antigen. So that's probably why they've actually teamed up to do this. And the whole inactivated vaccine is actually relatively easy to produce as well. So it's just easier. But some people may not respond to the mRNA vaccine. They may not respond to the adenovirus vector vaccine, immunogenic to reactogenic. So the subunit and the whole inactivated vaccine may be much more effective in those people. And the whole virus inactivated vaccine actually is very good at producing a wide immune response to the virus, but you just get very sick when they inject the whole virus inactivated into you.
Sure. I think you may have already answered this actually, but would taking this vaccine provide some protection to other coronaviruses? And I think what you said was actually it makes them more virulent or affects you more. Is that right?
No. So that is just one possibility with the enhanced antibody dependent effect. Okay. But I think generally a lot of these antibodies that are induced have some relative cross-protective effect against other viruses. So that antibody-dependent enhancement effect kicks in when those concentration levels of that antibody drop below a certain thing with dengue and other viruses. So it's not just what antibodies you have, it's what level of antibody you have that reduces their neutralizing effect, and which then becomes an enhancing effect when they drop below a certain level. So I think that the cross-protective effect might be possible in the immediate post-vaccine period when those antibodies are high, COVID-19 antibodies are high, okay? But then later on, maybe months later, when those COVID-19 antibodies are lower, they might, in some people, might become enhancing for the other coronaviruses. Does that make sense? Yes. So it's the post-vaccine antibody waning that is the key issue about when you look at enhanced antibody responses, and we saw that with dengue and some of the viruses as well.
Sure. When are we likely to see safety data on 1,000 or 10,000 participants, and will this be in place prior to us rolling it out, or will that be down to other factors?
Well, I mean, no. So the rollout. So if Pfizer's safety data at the end of November is okay, and the vaccine is approved in the FDA, the high-risk groups are going to get it first. Elderly, the ones with comorbidities, maybe frontline healthcare workers. France wants to give it to taxi drivers because they have most contacts. So some people go for the high contact individuals. And unfortunately, they will be the guinea pigs. You will see how the virus vaccine responds in those people and the protective effect in those people before people like us who otherwise are mostly healthy are then offered the vaccine. So in fact, this graded distribution of the vaccine will tend to allow you to see what's happening in other groups when it comes to you. And that's just the way it is. I mean, I'm not eligible for the vaccine straight away because I'm not in that high risk group. But my parents are. My parents are 80 years old, so they'll be in that front line of vaccinees getting the vaccine first.
Sure. So I know we've sort of still got a lot of questions, so I think I'll pick the last two questions. So one of them says, I had someone ask me, is it safe to inject man-made foreign genetic material into humans? Can it cause problems such as mutate or affect our genetic material adversely or cause problems with protein synthesis? I appreciate this is the process of getting most infections, but what are your thoughts?
No, I don't think that's an issue because, in fact, we probably have. I'm trying to think of a virus vaccine that does that. You know, the HPV vaccine, the virus like that's not a natural construct. Okay, the HPV vaccine is actually very good. It's one of the best vaccines we have. It's relatively almost no adverse effects. It works in over 90% of cases to induce immunity, and that's actually an artificial construct. It's a virus-like particle, so it's a self enclosing sphere with virus proteins expressed on the surface, which are, I think they're probably synthesized rather than actually harvested from a live virus. I need to check that. But the VLP is not natural. It's an artificial construct. I think it's actually I think it's the outer shell of the virus or a similar virus that then folds naturally and expresses protein on the surface. So you're creating something that doesn't exist from some natural building blocks. But I don't think there's a risk of that perversion of cellular machinery in that way.
Sure. And I guess based on the data you've seen so far, can we justify giving the vaccine to healthy under 40-year-olds with the data we have to date?
Well, I mean, most of those participants are actually in the 18 to 55 age group. And most of them are probably relatively healthy, although there are some, comorbidity groups in there. So I think most of the clinical trial data is actually based on that 18 to 55 year age group, because some of them don't have the elderly age group. So most of the clinical trial data is in that 18 to 55 year age group. And yes, I think it'll be fine.
Yeah. And I guess one final question from me personally is, what does the future hold with COVID? Is this going to be something like our normal respiratory viruses that mutates every year and we're going to have to keep chasing vaccines? Is it going to be like polio, where we are going to eventually phase it out once the vaccine is there and herd immunity takes place? What does the future hold?
Oh, I think this virus will be like flu. This virus will be there. It's not going to disappear. It's going to be like flu, and every season they'll check whether the vaccine is changing, but it's more stable than flu. So whereas flu, you have to vaccinate every year to combat waning immunity plus natural mutation. This virus, there are maybe 500,000 sequences in databases, and each virus only differs by another virus by about 8 to 10 bases. They're not in the same place, but this is a very stable virus. This is a big 30 kilobase RNA positive sense virus. It doesn't mutate very much. So you might find that you might need the booster every 3 years instead of every year.
Brilliant. Fantastic. Julian, thank you.
The virus, I think, as a seasonal coronavirus.
Fantastic. Brilliant. Thank you. Thank you so much for your time, for us allowing us to pick your brains. It's been really, really, really useful. So next we have Leah and Kay from the Leicester City CCG, who I understand will be talking a bit about the practical aspects of how we're going to implement the COVID vaccination program here in Leicestershire. So do we have Leah and Kay here? Okay. If we don't, Mayo, is there anything you want to share on behalf of West Leicestershire? I think you're just on mute, so just if you could unmute, that would be fab.
So I just wanted to make some remarks, both in my position as a national leader and also I lead the NHS Midlands region board for COVID and flu vaccination. So if the other staff had been able to join, they would have given you the exact details of the Leicestershire plans, but I'll be able to outline them for you.
Thank you.
So as you know, the NHS in England is divided into 9 regions, And the vaccination campaign for COVID is a very high priority for the NHS. And I want to reassure people that there is extensive planning and medical input into rolling out the vaccination. Obviously, this is all predicated on an approval for a vaccine, but based on the information you've seen and heard from Dr. Tang, I think you can anticipate that this will be addressed very, very quickly. I appreciate that a lot of questions and uncertainties about vaccination, but I'd like to remind everyone about how toxic and deadly this virus is, particularly in Leicester and Leicestershire. Currently, our wave 2 is probably more severe than wave 1. And overall in the country, 53,000 people have died. So this is a very, very important development. And I guess my first message to everyone on this call, and the fact you're joining us on a Friday evening, is that we have to engage and prepare for a rollout of vaccination and actually help people deal with the uncertainties and complexities. This is what general practice is for, isn't it? We're used to dealing with mixed information and supporting our patients. So this morning I had the privilege to chair the Midlands board, and we looked at the plans for all areas in the Midlands. So that's 11 what's called integrated care systems or partnerships with multiple PCNs. In Leicestershire, we have 25 PCNs, and every area has a strong plan for the rollout of this vaccination. So we have got this covered, and a lot of the practical things that Julian and others have mentioned, like storage, temperatures, the logistics, have actually essentially been formulated, and I want people to know that. So the medical director of this program in England is actually a GP, Dr. Jonathan Leach. He's in Bromsgrove. I know him personally. I've worked with him at the college, but he's also got experience of logistics in the military. So they're very used to doing mass campaigns. So let me just briefly outline what the model will be. So it'll be a national call and recall, supplemented by practice call and recall. There'll be a lead provider model in each integrated care system, working with PCNs, each of whom will have designated a site for the vaccinations. And most places have now got a provisional list or idea of their vaccination center. This is subject still to regional and national approval, so I can't actually announce what those are. Everyone will have a choice whether they go to a local primary care network center for vaccination or whether they go to a mass center, which might be a stadium or a hall or something like that. The next thing is there'll be a digital app, which is already being used in community services, for example, which will make sure that when a vaccination is given, it will be automatically uploaded to your GP computer system. This is still subject to confirmation and rollout, but such an app already exists. Now, a couple of things to respond to that came out on today's discussion. There'll be a national communications campaign. Obviously, we appreciate that there's a lot of anti-vaxxers social media out there, but we want to be honest and transparent. This won't be coercive. We have to be upfront with people, and it will be trying to persuade them using science and evidence. And but from my experience of talking to people, I think generally people are very keen, and there'll be a hotline as well, staffed by clinicians. So a couple of things to clarify just as I end. One is that the 15-minute observation period is not required now unless somebody's driving, so that makes it much easier. And the logistics about temperatures have been sorted out, so this will be delivered to you in the state where you can keep it at temperatures 2 to 8 degrees. It has to be used fairly quickly, typically within 5 days, hence the 7-day window, so the vaccine wastage is very limited. There'll be a list of vaccinators in each PCN or area who would go in and do this. The vaccinators will need very brief online training. I'm not sure the extent of what the requirements will be, but it will be very, very nominal, and there'll be national PGDs. Housebound patients will be a priority, and perhaps practices will be required to do that first. So there is a list of people who are eligible for vaccinations. There are 8 or 9 groups that's already been published. It's in the public domain, including doctors and nurses. So finally, let me end by saying that flu is still a priority, because there has to be a gap of 7 days between flu and COVID. And between the 2 COVID vaccinations, it will be 28 days. So your sequence might be FCC, so flu, COVID, COVID. I think that's the most ideal arrangement. But I would remind you, and everybody has your own practice data. If you don't know your practice data, ask your manager or partner for it. Every practice in this country has its own flu data, right? And I can tell you that many patients at the at-risk group or in care homes have not yet received their vaccinations. And that will leave them vulnerable to COVID infection and may delay COVID vaccination. As you'll know from the papers in BMJ, a concomitant infection with flu and COVID is a double whammy and increases mortality. So the ideal scenario is flu, COVID, COVID, 7 days, 28 days, 28 days. If you haven't had your flu jab, then you can have COVID, COVID, flu, CCF. So COVID, 28 days later, COVID, then flu 7 days later. So it's really important that you don't kind of think it's either or. And of course, the 50 to 65-year-old campaigns are starting. So can I just end by saying I really want your help in engaging with this. Be prepared to volunteer, in inverted commas, to be part of the vaccine workforce. Be prepared to dispel the myths that people will have concerns, to have honest and frank discussion. I'm certainly going to have the vaccine when it first comes. We'll be in the first cohorts, doctors and frontline nurses. Having worked on the COVID service and having done shifts this week, this is a really serious, dreadful illness. We know in Leicestershire today, Dr. Gershlik, one of the most eminent cardiologists in Great Britain, if not the world, died today from COVID. This is public knowledge, has been on the press. So, I appreciate this idea of, well, is this being rushed or not, but look, this is a public health emergency. If you have a choice between getting a virus or having a vaccine that's been approved, and hopefully it will all go through and is good enough, because it's all about choices, at the end of the day, it's about choices. And who better than doctors, GPs, to undertake shared decision making? So there are detailed plans for Leicester and Leicestershire and Rutland. I'm sorry my staff are not on the call today, but you will get that information. They will be published very shortly. So we have sites, we will have protocols, and there's a national DES already agreed, or sorry, enhanced services, not a DES. And there's a couple of sites that I would direct you. One is the NHS Futures Forum, which has a lot of detailed information. And there's national webinars being planned next week for the detail. So I'll stop there. Thank you, Abbas.
Thank you, Professor. A couple of you're undoubtedly going to get questions. So one question that I had. So about 2 or 3 hours ago, Matt Hancock announced that the vast bulk of vaccinations will no longer be done by GPs. It'll be done by vaccination hubs. Now, is that something that will apply in Leicestershire, or is he just talking?
Okay. So no, I think it's all about terminology. So the model we've gone for nationally is a lead provider model, because this vaccine is not suitable for individual practice delivery due to its characteristics, its fragility, and the need to, if you like, not waste vaccine supplies. Therefore, a mass vaccination approach is useful. It is my belief, and I think most people are involved in this, that the majority of people will be vaccinated through their GPs, through primary care networks. So it won't be through individual practices, yeah, necessarily, although just speculating, I think in the future, for other waves, it may be that we might, once the stability of the vaccine is established, for example, if it's a lyophilized vaccine that Dr. Tang said, then actually, you know, we could manage that, couldn't we? So it's urgent, it's quick, but I think the important thing is people will have a choice because we don't want there to be any delays. I mean, one example of the flu vaccination campaign is, you know, the uptake and the performance of general practice has been brilliant, much better than last year, but there's still an enormous variation and it's not quite, you know, so I think it's a different way of, it's a different mindset. Looking, let's PCNs do it, but also have mass vaccination centers, because remember all the IPC control applies. So I don't know what quite was meant by the Secretary of State, but I think probably what he meant was it'll be PCN led rather than individual GP practices.
Sure. Supposing within a PCN, individual practices can comply with IPC and the cold chain policy and ensure there's minimal wastage or not at all, is that something that PCNs can decide amongst themselves as long as they come to an agreement, all the policies can be followed, and we ensure that there's no wastage? Can PCNs then devolve the responsibility of that to practices?
Well, at the current stage, that's not going to be possible. I mean, I think it's important that I'm clear about that, not to raise false expectations. So the delivery will be to one site, the PCN designated site, and the vaccinations will be expected to be done there. Okay. You know, and there's a lot of reasons for that, which we probably haven't got time to go into. And I appreciate there's a debate to be had, but look, our trade union, the BMA, has agreed that arrangement. And, you know, I think I'd rather we get behind, because, you know, most of us are in PCNs, well, almost all of us, maybe there's one or two practices nationally that are not, but we do need to work together. You know, this will bring us together. And, you know, it's a national effort. It's like a wartime situation. You know, and the other thing that's come to me is the 7-day issue. The reason for that is, say if you get a delivery on Thursday or Friday of 975 doses, if you can't get through that, you don't want to waste that. This is precious life-saving vaccine. Therefore, the weekend arrangements are important as well to make sure you use all the vaccines.
Sure. Okay, brilliant. And I guess we've got the anticipated rollout on the 1st of December. Is that likely to happen on the 1st of December?
So I think it's more likely than not. I do think there's a high chance. I certainly think it's more than 50%. I think the delivery and the supply and logistics, that's exactly what's being sorted out. And obviously, as more vaccines come on board, this is where I think Julian's talk has been brilliant and fascinating. Then that will introduce a kind of more nuance into it around, you know, it's a bit like having different flu vaccines, isn't it? How do you match it to people, you know? So I would urge, you know, this is one of the things a lot of people, I've been talking about this for a while because of my role, but people are saying, you know, some people say, well, I don't believe it, it's not going to happen. You know, it takes years to get a vaccine approved, you know, the regulatory authorities are too slow, etc. But, you know, can I say, please do believe this. I'm very sure that we'll be starting as soon as possible, and certainly this year. And I think we have to be ready for that. And, you know, we can serve our populations. I appreciate, you know, we're very good at planning when things are uncertain. Look at the world's largest quality management program for long-term conditions, otherwise called the QOF, right? Nowhere else has that been done. Look at the complexity of that and the data collection. We did it, to the surprise of the Treasury, the government, and everybody else. We now have amazing world quality databases. So we can respond to it, but I think it does need that mindset and this thing around being ambassadors for prevention, rather than curing COVID, which is very difficult at the moment.
Sure. So Dr. Tang's got a question actually. So he's saying how given what he said about the adverse effects following the first dose, how much are we going to chase people for the second dose? Is this our role? And from what I understand from what you're saying, it's a national recall program through national call for first dose and recall for the second dose.
Okay. So there will be people who won't want a second dose because of a reaction they've had, or they might choose not to have it, providing there is a legitimate reason for that. And that wouldn't include, for example, that they haven't been called or for some other administrative reason. It has to be a clinical reason or a patient choice reason. You'll still be eligible, to the best of my knowledge, you'll still be eligible for your full payment because you've done your best to deliver the second dose, but because of a clinical reason, that's not actually happened. So that there is an exception for that. We've got to be very ethical about this and professional. So there's no coercion, or we can't just mandate people to have it. We live in a very democratic country, so I think that's very important. On the other hand, and can I say this is a really important point, it's a time for leadership from doctors. We can understand science and information. I don't want us to hide behind protocols and uncertainty and say, oh, we just don't know. Actually, it's balance of probabilities. But this is quite strong evidence. Other vaccines have been compared like this. We just haven't got the time. Anybody who's seen somebody with COVID, who's visited a COVID ward, or have got no doctors who worked in ITU, you'll think very differently about prevention then.
Absolutely, and I think that's one of the reasons why we held the meeting that we're holding this evening, is because we wanted the leadership there. We wanted clear up as many uncertainties as we can. So that's why we paired Dr. Tang's amazing scientific knowledge and background with your leadership and your expertise and skills. So thank you very much for your expertise on this. We've got a question about if a person is having chemotherapy, I presume they shouldn't have the vaccine as they won't respond. Will there be any risk to family members or spouses if they have the COVID vaccine? I presume that's more of a question for Dr. Tang than it is for Professor. Are there any questions, particularly in terms of the logistics of the rollout to Professor Mayo? No. Okay, so I think let's call it a wrap there. I know it's 9 o'clock, it's a Friday evening, and I know a lot of you have joined us since 6:45. And just echoing what Hina said, that your leadership is invaluable, your experience, in the past. As I said a couple of days ago when we did the leadership webinar, your experience in the past, present, and in the future is absolutely vital to keep general practice, particularly in Leicestershire, heading in the right direction. Yes, so we'll call it a wrap. So thank you very, very, very much to all of you for participating, for listening, for attending. I'm sure you've benefited a lot, just as I have. And a sincere thanks to our speakers, Dr. Julian Tang, Professor Mayur Lakhani. And I hope you all have a fantastic evening. Stay safe, and hopefully we'll see you soon.
Thank you. Bye.
Thanks.